In Chapter 5, we will discuss the process of photosynthesis, where certain living organisms can harvest energy from the sun and turn it into energy in the form of glucose. If you recall from Chapter 1, organisms such as plants, some algae, and some bacteria that absorb the sun's energy and convert it to glucose are referred to as producers or autotrophs because they can produce their own energy via photosynthesis. If an organism must consume energy to survive, it is known as a consumer or heterotroph. All energy available on Earth can be traced back to the sun. The radiant or light energy from the sun is used to drive photosynthesis, which also uses carbon dioxide from the air and water from the ground in combination with chlorophyll molecules in leaves to produce glucose and oxygen. This glucose is used by the producer as a food source, and the oxygen is a byproduct which we breathe. Photosynthesis has two phases: the light-dependent reactions, which require light particles known as photons, and it happens in the membranes of the thylakoids. This phase represents the photo portion of photosynthesis. The second phase, which represents the synthesis portion of photosynthesis, is the light-independent phase, as light particles are not necessary for it to proceed. This phase is also known as the Calvin cycle and the carbon fixation reactions phase because carbon is utilized to build glucose. This second phase occurs in the fluid-filled stroma, and it requires products from the first phase. Watch the linked video for an overview of the process of photosynthesis. Before we get into the process of photosynthesis, let's review a few basics of the cell and the electromagnetic spectrum. First, the location where photosynthesis occurs is in the mesophyll or middle cells of leaves. Inside the mesophyll cells are chloroplasts, which hold stacks of thylakoids. These stacks are called granum or grana for plural and are surrounded by a liquid called stroma. These two parts of the chloroplasts, the thylakoid and the stroma, will be important in the two phases of photosynthesis, the light-dependent and the light-independent reactions. Looking more closely at the chloroplast, notice it has a double membrane, both an inner and an outer membrane. The inner membrane encloses the fluid-filled stroma, which contains enzymes required to produce carbohydrates. Suspended in the stroma are the stacks of thylakoids, which are a third system of membranes. Inside the thylakoid is the area known as the lumen. It is in the thylakoids that the light-dependent reactions occur. For more detail on chloroplasts, click on the video link. This link will take you to a Britannica page with more detail and the short chloroplast video. There are two parts of plant cells that have similar names but serve very different functions and are in different parts of the leaf. These two parts can be confusing, so before we get any further, let's look at them. The stoma, without an R, means mouth and is a little opening on the exterior of a leaf where water and oxygen are released and carbon dioxide is taken in. Notice on the bottom on the left picture where the red arrow is. This area is also important in controlling moisture in the leaf, so it will close on very hot days to help conserve water. The plural form of stoma is stomata. The other cellular part is the stroma with an R, which is found in the chloroplast inside of the cell. This is a fluid-filled area and is the site of the light-independent reactions, or the Calvin cycle, of photosynthesis. The sun emits a range of radiation, and all of it travels in waves. Waves can be measured by their wavelength, which is from crest to crest or from trough to trough. The longer the wavelength, the lower the energy, and the shorter the wavelength, the higher the energy. Looking at the bottom image, left to right, the range of radiation is from very high energy gamma rays to low energy radio waves. Right in the middle is visible light, the only radiation we can see with the naked eye, and within visible light, blue and violet are the shorter, higher energy waves, while red and orange are the longest, low energy waves. The pigments in chloroplast absorb some colors while reflecting others. Colors that are not absorbed are reflected, and that is what we see in plants. Notice green is in the middle of the color spectrum. Most of the time we see green leaves. So what does that mean? Are green light waves absorbed or are they reflected? If you think reflected, you are correct. Chlorophyll readily absorbs the colors on the ends of the spectrum and reflects green. Note, when you see the yellows, oranges, and reds in the fall, that is due to a winterization process. Inside the membranes of thylakoids are molecules known as pigments which absorb energy from the sun. Notice in the diagram the little green circles. These represent chlorophyll molecules absorbing the light energy. There are two types of chlorophyll, A and B, as well as carotenoids. Chlorophyll A is responsible for getting photosynthesis started. However, each type of pigment absorbs different wavelengths of light and can contribute to the function of chlorophyll A. Chlorophyll molecules reflect green, which is why leaves usually appear green to us. The carotenoids reflect yellow and orange, and therefore we can see those colors in certain plants, such as a carrot or squash. Light energy is made of small particles from the sun called photons. Inside the thylakoid lumen, photons are absorbed by chlorophyll molecules. So now we're going to actually get into photosynthesis, and we're going to start with light-dependent reactions. So the term light-dependent means this phase requires light or photons. When the photon reaches the thylakoid membrane, it excites a molecule of chlorophyll, which releases an electron. The donated electron passes energy to other chlorophyll molecules until it reaches an electron acceptor. A molecule of water is split during this process, which replenishes the lost electron and allows more photons to be absorbed by the chlorophyll. When the water molecule is split, oxygen and hydrogen ions are released. These hydrogen ions will be used later in the second phase, the Calvin cycle. A more detailed look at the process of the light-dependent reaction illustrates the numerous steps in this process. Once the chlorophyll molecule has released an electron, it will travel through an electron transport chain similar to that in the mitochondria during cellular respiration, passing through photosystem II and then passing through photosystem I. As the electrons are passed through these systems, the hydrogen ions are pumped against the gradient into the thylakoid space. These hydrogen ions reduce NADP+ and ADP into NADPH and ATP. Remember, when molecules are reduced, they gain electrons and therefore energy. Just like in cellular respiration in the mitochondria, the electrochemical gradient created by the hydrogen ions will then drive the protein complex ATP synthase to reduce ADP and inorganic phosphorus into the energy ATP. When ADP plus is reduced to NADPH, it stores the energy and will be used to build a glucose molecule in the next phase of photosynthesis. Did you notice that electrons traveled through photosystem II before they traveled through photosystem I? This is only because PS1 was discovered before PS2 and are therefore named based on discovery and not on the order which molecules move through them. This image is a repeat of the previous slide, but may look similar to the image of the mitochondrion when we discussed cellular respiration in the previous chapter. Also note in this artist's depiction, the stroma and the lumen are flipped compared to the previous slide, but the concept is the same. A process similar to osmosis is chemiosmosis, and this involves moving ions, like hydrogen ions, across a membrane. You first heard about it during cellular respiration. Now, looking at the diagram, notice the blue arrows represent the flow of electrons from one protein to the next. These electrons start in PS2 and then go through the complex known as an electron transport chain. As these electrons move through the complex, they pump hydrogen ions from the stroma into the thylakoid lumen. After PS1, NADP positive is reduced to NADPH, which carries more energy. When the level of the hydrogen ions pumped into the lumen are greater than in the stroma, chemiosmosis will occur through ATP synthase. Just like in cellular respiration, these ions rotate ATP synthase. When the enzyme rotates, ADP and phosphorus create the energy ATP. Both NADPH and ATP are used in the next phase of photosynthesis to build the glucose molecule. The ultimate goal of the light-dependent reaction in photosynthesis is to capture light from the sun and produce two high-energy molecules, NADPH and ATP, for the light-independent reactions, also known as the Calvin cycle. During this phase, water and light are taken in while oxygen is released, and energy in the form of NADPH and ATP is produced. Watch the linked video for an animation of the light-dependent reaction. The first phase of photosynthesis requires photons from the sun to produce energy. This second phase is referred to as the light-independent phase as it does not require light. It is also known as the Calvin cycle after the scientist who discovered the process. In this second phase of photosynthesis, the energy produced in the first phase is used to take inorganic carbon from carbon dioxide and convert it into an organic carbon molecule like glucose, which can then be used for food. This inorganic to organic conversion process is known as carbon fixing. In the Calvin cycle, plants take in carbon dioxide, which enters the stroma of chloroplast. This part of photosynthesis has three main steps. Number 1, carbon fixation, 2, carbon reduction, and 3, RUBP regeneration. It requires one full turn of this cycle to process the one carbon atom of carbon dioxide. As a result, to process a 6-carbon glucose molecule, the cycle must repeat a total of 6 times. This is a complex diagram on the left of how 6 carbon dioxide molecules are taken into the Calvin cycle. You will not need to know all of these steps, but rather just understand the big picture and the 3 main steps as noted in the previous slide, which are numbered in the center purple section of this Calvin cycle image. With the help of an enzyme, Rubisco, a carbon atom from carbon dioxide is joined to one molecule of RUBP, creating an unstable 6-carbon molecule. This 6-carbon molecule is broken into 2 3-carbon molecules called PGA. Notice these first 3 changes in the Calvin cycle are illustrated in the diagram carbon fixation to the top right. The next step of the Calvin cycle is the carbon reduction phase, where these 3 PGA molecules gain an electron, are therefore reduced, and are now called G3P. This second part requires ATP and NADPH, which were products of the light-dependent portion of photosynthesis. The ATP and NADPH are oxidized, which means they're losing their electron or energy, and are then returned to the light-dependent reactions to be reduced to gain an electron and continue the photosynthetic cycle. 6 carbon atoms are ultimately pulled out of the cycle to form one 6-carbon glucose molecule. The remaining carbon atoms and ATP are left in the cycle to regenerate RUBP to continue the Calvin cycle with future CO2 molecules. For a summary of the big picture, Step 1. Carbon from carbon dioxide is fixed from inorganic to organic 3-PGA. Step 2. 3-PGA is reduced, meaning energy is added, to make G3P, where then carbon is pulled off to be used for glucose synthesis. And finally, step 3, the remaining carbon atoms will continue in the cycle to produce more RUBP for future carbon fixation. Watch the linked video for an animation of the Calvin cycle. Plants living in hot and/or dry conditions have special structural and physiological adaptations that help allow photosynthesis to continue even when carbon dioxide is limited. Stomata will stay closed during the day, reducing water loss, but then open at night so carbon dioxide can enter the stroma and photosynthesis can occur. For prokaryotes, which were cells lacking a nucleus, that are autotrophic and therefore perform photosynthesis, they do not have chloroplasts like plant cells. These cells have chlorophyll molecules that can capture light, and these chlorophyll molecules are found in the folds of the cell membrane. Although the location of photosynthesis differs between prokaryotic and eukaryotic cells, the process of capturing light and converting it into carbohydrates is just the same. Plants can produce glucose through photosynthesis, but they can also perform cellular respiration to break the glucose down into a usable energy form. Plants release oxygen and need carbon dioxide, while animals need oxygen and release carbon dioxide. Between the two processes of photosynthesis and cellular respiration, there is no waste of materials. Molecules are recycled in nature as shown in the image. This is the end of Module 1, which covered chapters 1 through 5 and the first 4 labs: microscopy, diffusion, fermentation, and photosynthesis. On Friday at 12 midnight, the exam will be available to you. It will be available until Saturday night at 11:59 p.m. Once it is opened, you will have 90 minutes to complete the test. You cannot save and return. It has to be completed in one sitting. The test is 45 questions for a total of 50 points. There are multiple choice questions, matching, and one fill-in-the-blank question. To review for this test, go back and look at section headings in the text, go through the end of chapter review questions, browse the lecture slides, and review the material in the handouts.